Okay, hello everybody out there in cyberspace. Happy Friday afternoon. I know you've been waiting for this lecture and everyone's been looking forward to talking about respiration, so let's get right into it. This week in the lab, we're talking about fermentation, which is the point where the labs have slightly got ahead of the lectures. After this lecture and the next one, hopefully the lab will make sense. If you're not so familiar with fermentation, we're not going to actually get to fermentation today. This is a two-part lecture. Today we're going to talk about glycolysis, the citric acid cycle, and maybe we might get to that electron transport chain. So this is about where we left off. We were talking about enzymes and we were talking about cellular work. And I had said, this is why we need ATP. So what kinds of cellular work were we talking about? We were talking about things like transporting things across membranes, moving things around cells, or even moving things like your muscles, if you're an animal, moving things like flagellas. And lastly, we talked about something called energetic coupling or energy coupling, where we're using ATP to basically build new chemical bonds. This all takes a lot of ATP. If you are a muscle cell, you're using about 10 million ATPs per second. Unless it's per minute, I can't remember. It's still a lot of ATPs. Yeah, so especially as animals, we need to be making ATP all the time. So how are we going to do this? We're going to consume food. We're going to break it down. We're going to take energy from the bonds in the food, and we're going to make ATP. So today, that's what we are going to talk about. How do we make ATP? We phosphorylate ADP. So remember, ADP is adenosine diphosphate. Phosphorylation means we're going to add a phosphate onto something. And here's our little phosphate, an inorganic phosphate. So we're going to take some energy. And with that energy, we're going to make ATP. This is the molecule we want that's going to help us to power all that cellular work in the cell. So how do we make ATP? So a little bit of biochemistry for you in terms of, these are things you're going to talk about in your second year if you take a biochemistry class. But there's technically two ways that we make ATP. One of them is called substrate level phosphorylation. So what does that mean? It means that the phosphate group is going to come from another substrate. So that's what we are seeing right here. We've got some sort of carbon molecule and it's got a little phosphate attached to it. And then that phosphate is going to detach and it's going to go attach onto ADP and we're going to get ATP. So that's one way we're going to do that. You're going to see that in glycolysis and you're going to see that in the citric acid cycle. And we're not going to be able to make a lot of ATP that way. If you want to make a lot of ATP, this is the way to do it. Oxidative phosphorylation. So you may have heard about the electron transport chain. Oxidative phosphorylation is part of that. So this is going to use molecules like this, NADH. We're going to think of that as an electron carrier. Excuse me. And that electron carrier is going to bring those electrons to the electron transport chain. A whole bunch of interesting chemistry is going to happen. And in the end, we're going to take this phosphate and this ADP. We're going to put them together and make ATP. So I'm not sure if we're quite going to get to that today, but we'll see where we get. So substrate level phosphorylation, like I said, is just transferring a phosphate. So here's a phosphate and it's got a little bond and this bond is just going to go over there to the ADP and we're going to get an AT. Oxidative phosphorylation is going to occur in the mitochondria. It's going to involve all these mitochondrial membranes. We're going to have some chemical gradients and things like that. So it's a bit of a different process. So like I said, more on that later. Maybe we'll get to it today, maybe it'll be next day. So some other terminology for you when we talk about respiration. Sometimes we talk about these redox reactions. So you may remember oxidation and reduction reactions. So an oxidation is where we lose electrons. You can see we have a little compound here. It's got some electrons and those electrons are gone. And so that's an oxidation reaction. The other way around, this molecule here, B, gains those two electrons, so it becomes reduced. So this is a little thing. I think other people have learned this one as well. This little moniker, I learned it from my high school teacher. And it says, Leo says grr. So Leo the lion says grr. So Leo means loss of electrons, oxidation, gain of electrons, reduction. So one way to help you kind of remember which is which. So it's pretty easy to think of oxidations when you are talking about this in chemistry class and you're thinking about electrons. So, you know, you can see here we have sodium chloride being produced. So we have this sodium, right? And sodium has, you know, an extra electron sitting around here, and your chlorine has not quite a complete octet, right? It's got 7 in its octet. And so, hey, it would be just nice and convenient if one electron would just flip over, and that's what's happening here. That electron is getting removed from sodium, stripped off, and ending up on the chloride. But this is kind of like, you know, one of the problems with teaching chemistry the way we do it is electrons rarely kind of do these kind of things, just float around by themselves. And even more rare for them to do things like that in a biological context. So what is happening in a biological context? So this is methane, and I'm just showing you this as a very simple organic molecule. And just take a look at methane for now, and you may notice we've got our carbon, and our carbon has an octet, right? That means it has 8 electrons. Each of those electrons are in pairs, and each pair of electron represents a chemical bond. So just take a look at this one here. There's a chemical bond of carbon and hydrogen, 2 electrons, 1 chemical bond. So in biology, we have to think of oxidations as involving bonds, but it also involves how those electrons are shared. So take a look at, you know, carbon and hydrogen, they share the electrons equally, right? So, you know, that's not too bad. They kind of like it that way. We've got an oxygen molecule, and it's a double bond, and it's sharing its electrons equally. But watch what happens in this oxidation process. So we can burn methane with oxygen, and we get carbon dioxide. So here's carbon dioxide. And notice that oxygen and carbon are sharing the electrons, but oxygen is not sharing fairly, right? Oxygen is very electronegative. So what has happened here is that carbon has become oxidized, meaning it doesn't have as many electrons near it as it would like. So this is kind of what an oxidation is like in biology, where we're talking about carbons and the state they're in. And if you think about things like formal charge and all those things that you learn in Chemistry 101, this is kind of what it's all about. So just remember, oxidation usually means you're breaking something down, and often you're using oxygen, but not always. But usually something is getting broken down and it's losing energy. This is kind of the key thing, right? Oxidation usually means something is losing energy. So the carbon, the methane is losing energy. So something to think about. Okay, so here's the oxidation reaction we are going to look at, which is the breakdown of glucose. So we're going to break down glucose, which is 6 carbons. You can see C6. We're going to break it down into 6 carbon dioxides. We're going to oxidize it with oxygen. We're going to make carbon dioxide water, and energy is going to get released. And that energy, as cells, we're going to try to capture as ATP. So as I mentioned, oxidation reduction reactions in cells are usually associated with chemical bonds, and often we're actually associating them with hydrogens. And by hydrogens, I don't just mean the nucleus. I mean the hydrogen and the electrons that are attached to it, right? So if you think of, like, I had my methane here, right? So here's my methane. Draw that out for you. And my methane is a hydrogen plus 2 electrons, right? So that's what we're talking about. So when we remove that hydrogen and usually electrons, we call that a dehydrogenation. So you can see D for removal, hydrogen because we're removing a hydrogen, and then ation meaning, hey, we're just turning the whole thing into a verb, right? So when you see these terms, oxidations in biology and dehydrogenation, they actually kind of usually mean the same thing. So there's an example of an organic molecule on the bottom. You can see over here we've got a carbon. It's attached to some different atoms. It gets oxidized, and we have NAD+. I'm going to talk about that in a moment. Is going to gain the hydrogen. So you can see it becomes NADH. And also it gains the electrons that are attached to that hydrogen, so it becomes reduced. So I want to take a minute and talk about this NAD plus thing, okay? So this is an electron carrier. So what do I mean by that? So here's NAD plus, by the way. You don't need to know the full word. It stands for nicotinamide adenine dinucleotide. So that's why we call it NAD. It's much easier just to say NAD. So this is actually derived from niacin, which is like, I don't know, vitamin B3 or something like that. And if you take a look at the rest of the molecule, we have, oh, what is this? This is adenosine. These are a couple of phosphates, and here's another ribose. So this is just one of these nucleic acids that's not involved with DNA. It's involved in energy stuff, right? So the nicotinamide part is shown in the box, and I want you to take a look at the nitrogen there that's shown in blue. It has a positive charge. So that's why we're saying NAD+. It's an abbreviation. So we eat our apple or whatever food we're eating, it gets oxidized. Electrons, so here's the electrons, and hydrogens, not loose, they're going to be wrapped up in bonds, probably together, end up attaching to our NAD+. So there it is, we got a new hydrogen, a new bond. So NAD+ becomes NADH. So I like to think of it as NAD and then bond H. And that helps me remember that that H actually means hydrogen, and it's connected there by a couple of electrons, and it's the electrons that are important because they carry energy. So I think this kind of has a summary on just some arrows, and there's the overall reaction. So anytime we see NAD+, it's going to go in, it's going to oxidize something, and this NADH here is going to be a very important molecule because it is going to go to the mitochondria. And we know good things happen in the mitochondria. We're going to make lots of energy in the mitochondria. Okay, so there are other electron carriers out there. And we're going to see one more, actually two more in this class, but one more today. And this is called FAD. So if you look at it, it's another fancy molecule. It's got basically another nucleic acid here. Okay, and we've got flavin instead, and that's I think some sort of other vitamin. I can never remember. There's too many of them to remember, but it's part of your diet, and it also is going to get reduced. So it's going to oxidize some sort of food product, and it's going to take those electrons, and it's going to also take them over to the mitochondria, and in the mitochondria, good things are going to happen. We're going to make lots of ATP. Okay, so back to kind of our summary. How do we make ATP? We phosphorylate ADP. We can do it in a couple of different ways, and we're going to see both of those ways in lectures for topic 10. Okay, so maybe before I get to the slide here, I'm just going to, let's see here. Bring up the whiteboard. I'm going to find the whiteboard. And I'm going to write a couple of notes for you. Okay, I guess I got to share the whiteboard. Find Zoom. Well, there's our whiteboard from last day. So let's just go here and we'll make a new one. Okay, so first thing is, I know people are thinking about, okay, you maybe saw glycolysis and the citric acid cycle and all those things before somewhere. What do we need to know? Do we need to know every structure and every enzyme and all that? No. Here are things that I want you to pay attention to. So we'll just call this one things to know. Things to know for metabolic pathways. Okay, so this is what I want you to pay attention to. Okay, I want you to pay attention. What is happening to the carbon? Okay, and number two, I want you to pay attention to what is going on with ATP. Where are we making it? Where are we consuming ATP? And then number three, I want you to pay attention to those electron carriers. Okay, so for example, let me see if I can Just going to move this over here. Hopefully we have enough space to do this. Let's just talk about glycolysis for a minute, and I'll go back to the slides, right? So glycolysis starts off with glucose. Okay, so glucose is a 6-carbon compound. Maybe I'm going to make that just a little bit longer. How do I erase that? Oh, I could just erase. There we go. Make the arrow just a little bit longer. And we're going to make 2-pyruvate. And just need to move it over just a little bit more. There we go. So there's our carbon, right? Pyruvate is 3 carbons. Okay, so I'll just say this right here. We've got glucose, we're making pyruvate. Obviously, there's some details in the middle, and I'll try to point out what is kind of the most significant. Do I care that we're making 2-pyruvate? I care. Do I care for this course? Not so much, okay? I know that some people are really fond of balancing chemical equations and things like that. I'm not so concerned about people being able to balance chemical equations. That's for another class and another time. It was great if you can count. Good job there, but it's not really that important. What I care is you know, the overall picture of what we're doing. So we're breaking down glucose, right? So glycolysis, glycolysis, right? So glyco means carbohydrate, lysis means breaking. So we're literally breaking glucose into 2. It's not that simple. There's a few other things going on here, but in a sense, 6 carbons to 2 3-carbon molecules. So the other thing to consider about glycolysis is that we are making 2 ATP net. We're actually consuming some in there, but so overall net. So to make ATP, we're going to make 2, by the way. This takes 2 ADP and 2 inorganic phosphate. So there they are there. And we're going to make 2 ATP. And the last thing I told you to pay attention to are those electron carriers, right? So the most common electron carrier is NAD+. We're going to take 2 of those, and we're going to make 2 NADHs. So that is kind of, in some ways, it. Okay, not quite, but this is, for glycolysis, you know, a good 75% of what I want you to know. I want you to know what's going on with the carbon. It's getting broken down. You know, we have words for that. Catabolism, right? You know, broken down. We can say oxidized. We're making 2 ATP. How do we make ATP? We need the ingredients for that. We need ADP and inorganic phosphate. And we're making 2 NADH. And what do we need for that? We need to oxidize a compound, and we need NAD+. So like I said, this is kind of 80% of what I want you to know about glycolysis. Everything else is just details, of which most of them aren't so important for this class. So focus on this. I'm going to come back to this, and I'm going to add eventually the citric acid cycle and a couple of other things in there. So if you didn't get all this, don't worry, I will come back to this, okay? So let's go back to our screen share and find our, gotta find the PowerPoint. Where did it go? Okay, here we go. There's the PowerPoint. Oh, I guess I just lost it. Oh, here it is. No, that's not it. Where did it go? I don't know. Sometimes you bring things up and then you can't see them on Zoom. Oh, there it is. Okay, I had to just minimize Zoom and bring up Zoom. Okay, so let's take a look at this. This is kind of the nice colorful version of what I was talking about in the textbook, and maybe a little less detail, and we are going to see a lot more detail. So you can see the very first step we're going to talk about is glycolysis, breaking down glucose to pyruvate, and we're going to make 2 ATP by substrate level phosphorylation. The rest of the processes are going to be happening in the mitochondria. We're going to actually break down respiration into 4 processes. We're going to break it down into glycolysis, pyruvate oxidation, the citric acid cycle, and then that other one that's not on there yet, which is oxidative phosphorylation. So pyruvate oxidation, right? Oxidation, we're breaking something down. We're going to break it down to acetyl-CoA. the acetyl-CoA is going to go into the citric acid cycle. So some of you might be thinking, citric acid cycle, what is that? Depending on the age of your high school teachers, right? Some of them may be calling it the Krebs cycle. Most of the textbooks have renamed it. So quite a while ago now too. So if your teachers still use the Krebs cycle, they're old school or they're just old. You know, relatively speaking anyway. So we're going to talk about that. Last part is oxidative phosphorylation. So that's part 4. This is going to involve the electron transport chain and chemiosmosis, which is formation of ATP. And we will get there, whether today or next day. So let's take a closer look at glycolysis. Glycolysis, you can see this is just a bit more detail than I showed you before, right? Has the same beginning, glucose, the same end, 2-pyruvate. And look, there's water. But did I tell you to be concerned about water? No, nobody cares about water. Well, our cells care about water. Plants love water. But really, when it comes to chemical reactions, it is so not important because water is everywhere around cells. So whether it's made or consumed, it's really not consequential at all. So notice it's breaking down into glycolysis into 2 stages, right? 1 here and 2 here. So the first stage is an investment stage where we have to actually add some activation energy to get the whole thing going. And that is actually going to consume 2 ATP. But in the end, in part 2, in the payoff stage, we're going to produce 4 ATP. So if you add all this up, the net is we are going to make 2 ATP overall once you take the 4 and subtract 2. So down at the bottom, you can see what they're doing here is basically summing everything up. And so, you know, that's mostly important. Like I said, we don't need to worry about this, water here. And we're not going to think of electrons as floating around by themselves. So we can kind of just get rid of some of these things here. And that's kind of most of the important information that you need to know for Biology 107, is kind of keeping track of those crucial things. So the carbon, the ATP, and the electron carriers. So let's jump into this. Here's glycolysis. There's glucose. Glucose is a great molecule. Tastes good, you know, nice, easy to draw, and has a lot of interesting things about it. There's a good reason why glucose is maybe one of the most abundant biological molecules on the planet. It's part of cellulose, it's part of our food, and there it is. So let's break it down. We're going to start breaking it down by throwing in a little bit of activation energy. So remember last day we were talking about how reactions, you know, they just sometimes need a little push, right? And so that's what we're doing to break this down. Sometimes it takes energy to get something out of it. So the very first step, you can see the ATP is being consumed and the phosphate is being left on the glucose. We get glucose 6-phosphate. And then we're going to switch things around a little bit. So that's called an isomerization reaction. We're making an isomer from glucose 6-phosphate to fructose 6-phosphate. So why do that? Well, it turns out that fructose is easier to break than glucose. So it's an easy step, you know, you just jiggle things a little bit and then you're almost ready to go. We're going to add a little bit more ATP to this whole thing. And now we have fructose 1,6-biphosphate, sometimes called fructose 1,6-diphosphate. So by the way, there is our little enzyme that we talked about last day, phosphofructokinase. So that is a very key regulatory enzyme in glycolysis. And maybe just as a reminder, I will write a note about, so I'm not going to write up the full name, it's very long, but phosphofructokinase. So remember what we were talking about last day is we had said that phosphofructokinase is negatively inhibited by PEP. So what is PEP? We're going to see PEP in a moment, which is phosphoenol pyruvate. So there's pyruvate there. It's got a phospho on it. And what's an enol? So if you take organic chemistry, you learn that an ene is a double bond and an ol is an alcohol. So somebody just naming this molecule and kind of describing it as it is. Well, we can call it PEP. It's also negatively inhibited by ATP, which is an end product to this whole reaction, right? We are making ATP. If we have lots of ATP, we're going to shut down glycolysis. We don't need more. But if we don't have a lot of ATP, then we probably have a lot of ADP, right? If our ATP is all broken down, we're going to have a whole load of ADP. So if we have a whole load of ADP, we want to turn glycolysis on. So it's positively regulated by ADP. So that's my kind of brief, short note. Remember last day I made you a much longer kind of detailed thing. If you didn't get all that from last day, just ask me. I think I still have it on the whiteboard. I can probably export it into an image file, and I can share that with you very easily. Let's move on with glycolysis. Energy investment. Put in 2 ATPs. We haven't done much yet other than added a couple of phosphate groups. But now that thing is primed. It's ready to go. It's got gasoline into the lines. We're ready to break this thing down. So here we go. Boom. We break it down into 2 3-carbon compounds. So what are these 3-carbon compounds? So you can see that we have one here called dihydroxyacetone phosphate. And we have another one here called glyceraldehyde 3-phosphate. So these two are kind of interchangeable. Very easy to change them back and forth. I want to point out this one here though. Glyceraldehyde 3-phosphate. So why is that one important? That one is important because we are going to talk about photosynthesis next week. And that is actually the end product of photosynthesis. So I know everyone's thinking, photosynthesis, don't we make glucose? Well, yeah, sometimes. Well, not we, but the plants sometimes make glucose. But some plants don't. Some plants will make fructose, some plants make sucrose. There's a whole variety of carbohydrates they can make. Glucose is just kind of the number one, right? They all make glyceraldehyde 3-phosphate. And you can see that these reactions, I could run them backwards, right? We can run this backwards. I can make fructose 1,6-5-phosphate. I can make fructose 6-phosphate. And I can just keep on going backwards. And I can make all these things very, very easily from glyceraldehyde 3-phosphate. But that is the product of photosynthesis. So I'm going to remind you again when we talk about photosynthesis. But just pointing out that there's another chemical name to learn. So on this slide, what do we need to know? Well, you should know the word glucose, I hope. Hopefully by now you're going to remember phosphofructokinase and then glyceraldehyde 3-phosphate. And we don't really care so much about dihydroxyacetone phosphate. It's kind of doesn't come up again. Okay, so let's keep breaking this thing down. So there is, just from the previous slide, continuation of that. And you can see we are doing a oxidation. So if you take a careful look at that compound, there's a whole bunch of things going on. It doesn't maybe look like an oxidation because we're doing some sneaky things in there. We're throwing in an extra phosphate, and there's some water molecules that aren't accounted for in that reaction. But we are getting an oxidation, and we are getting those electron carriers. So we're getting NADH. So that's good. We're making 2 NADH, right? Why 2? Because everything here is multiplied by 2 now, because we broke glucose into half, right? So everything's just kind of double. We're going to make some ATP. Look at all these phosphates kicking around. So we got a phosphate here. We can make use of it, and we can attach it onto this ADP. Boom. And there we go. Now we have ATP. Well, we have 2, because like I said, everything's multiplied by 2. So at this point, we have made a net of 0 ATP, because we consumed 2, remember, and now we've made 2. Don't worry, we are going to make 2 more. So here we go. We have 3-phosphoglycerate. And a couple of reactions happening. You can see a mutase, that's kind of like an isomerase. We're going to lose some water, and we're going to get, here it is, there's PEP. Okay, that's a regulatory molecule. And then the last step is we make pyruvate by stripping this phosphate off and putting it onto that ADP and getting ATP. Okay, so looking at these reactions, right? There's not a ton here new that I've added. I've already said ATP, we've already talked about pyruvate, we've already talked about phosphoenolpyruvate. So like I said, knowing the basics of glycolysis is what we need to focus on here. And everything else, these details, like I said, I'm trying to point out the important details as we go along. If you want to learn all the reactions, go for it. That's great. If you take a biochemistry class, you'll probably have to learn them anyway. So save yourself some time by learning them now, but you're not going to need to know all of them for this class, just kind of the overarching what is going on here. So there is kind of the payoff stage, all seen in one picture. And you can see we're producing a bunch of things, some NADH, some ATP, and some more ATP. So awesome, this is good. We're getting some energy, and energy is good. Cells like it. So there's back to that summary slide, right? And like I said, you know, most of the things on here are good to know. We don't need to worry about electrons because they're in bonds. We don't need to worry about loose hydrogens. Who cares about those? We care about our carbons, our ATPs, and our electron carriers. Don't worry about water. Water is important, but really in the grand scheme of things, the cells have lots of it. Okay, so that's glycolysis. 10 chemical reactions to get from glucose to pyruvate. We've made some ATP, we've made some NADH. And at this point, kind of depending on what organism you have and what you are, if you have oxygen, you're probably going to move things into the mitochondria. So that's actually what's shown here. We have pyruvate moving into the mitochondria, and we have a whole bunch of things that happen simultaneously. There's actually one massive enzyme that does this thing. It's called pyruvate dehydrogenase. We're going to call this reaction pyruvate oxidation. So this is kind of like step 2. It's separate from glycolysis because it doesn't always happen. And it's separate from the citric acid cycle because there's no cyclic component to this. So sometimes people do group it together with the citric acid cycle. So you can see what's happening here. Overall, we've got this here is a carboxylic acid group, a carboxylate group because it doesn't have the hydrogen. And it is basically getting ripped off, and basically now we have carbon dioxide. So what do we do with the carbon dioxide? Well, if you're a mammal, then you're done with it. You're going to probably exhale it. It's going to go into the atmosphere and get used by plants. We are also making an NADH. Okay, so that's good. We're oxidizing this whole thing. And the final product is acetyl-CoA. So acetyl-CoA is a good thing because we can use it for the citric acid cycle. So I just want to talk a little bit about this thing here, this coenzyme A for a minute. Okay, and I know I have a nice picture of it here. Here's coenzyme A. So what do we have here? We have yet another nucleic acid, right? In fact, it's adenosine all over again, except for there's a little phosphate down here. I'm not sure what it needs to be doing down over there. And then we've got this kind of all these other functional groups attached to it. So this is called coenzyme A. Often it's abbreviated as CoA-SH or CoASH. So the SH part is actually sulfur and hydrogen. You can see them right there, sulfur and hydrogen. So what are we doing with coenzyme A? You can think of this as, okay, you've got, you're breaking down pyruvate. That's not a lot of carbons. We want to make sure it's getting to the place it needs to get to. So you can think of coenzyme A as an escort or something like that, where it is carrying carbon, okay? In this case, just a couple of carbon atoms. If you take a look at, oh, there's some diagrams. I've already said that. That's adenosine. So if you take a look at this, right, there's coenzyme A at the top, or CoASH. And then this here, let's see if I have it labeled. I do have it labeled, is an acetyl group. So the acetyl group used to be on pyruvate. An acetyl group, you know, if you're interested in the chemistry, it kind of looks like this. We got a CH3. And then we've got a carbon that's double bonded to an oxygen, and then it's attached to something. So that's an acetyl group. If this were acetic acid, it would look like this, right? It would just have an extra OH on the end, and that would be an acetic acid. But we're just looking at the acetyl group, so we're not going to worry about that. So acetyl-CoA is just this CoA thing, okay? And we've added this acetyl group onto it. And that's just going to, like I said, it's kind of like it's just going to get escorted, you know, all the way through the mitochondria until it finds the next enzyme. And that's about it. By the way, fat molecules are broken down using acetyl-CoA, and it's involved in a whole bunch of pathways. It's just kind of a handy thing in the middle that, like I said, it's very easy to think of as an escort. Okay, so there's our pyruvate oxidation, and so now we want to talk about the citric acid cycle. You can still call it the Krebs cycle if you like. I know people, you know, are really fond of that word, and maybe, you know, you're a friend of the Krebs family, I'm not sure, but you can still call it the Krebs cycle, I don't mind. So let's take a look at what is going on in the mitochondria. Okay, so there's our little mitochondria. I know I showed you this picture before, and we're going to come back to it. Remember that it has two membranes. We've got an outer membrane and an inner membrane. And this inner compartment here, I'm going to color it in here, that is the matrix. And that is where the citric acid cycle is going to happen, right? So, by the way, if you ever, there's an old movie called The Matrix. Notice it's back on Netflix again. It's a good watch if you're interested. Okay, let's talk about the citric acid cycle. So up at the top, you can see pyruvate oxidation and everything you need to know about pyruvate oxidation. We've got a pyruvate, it gets broken down into acetyl-CoA. We've got some NADH being made, and we've got some carbon dioxide being given off. How do we make a CoA thing? We add in the coenzyme A. So that's kind of everything you need to know about pyruvate oxidation. Let's take a look at the citric acid cycle here. Or actually, you know what I'm going to do? I'm going to go back to my whiteboard and put that on the whiteboard. Little extra reinforcement for you so that you can remember all this stuff. Okay, so let's see here. How far can I? Can't shrink much further, can I? That's too bad. Let's see here. Can I? Maybe I can. There we go. Okay, I'm going to drag this over here. I can't shrink anymore, but it looks like I do have lots of space over there. So we'll see what I can fit on. So let's actually draw a membrane. Or actually, I guess this would be a double membrane, but I'm not going to do two dotted lines. It's hard enough doing one. Okay, so what do we have here? Here we have the cytoplasm. And here we have the mitochondrial matrix. Mitochondrial matrix. Okay, so there's glycolysis. It's cruising along out there in the cytoplasm. We get pyruvate moving across the membrane. Okay, and so how do I just need to drag this over a bit more. So I'm going to have to move glycolysis over. Sorry, oh, maybe I can shrink it down a little bit, just a little bit. I think I can just squeeze this on. So there's our 2-pyruvate, and I am going to balance my equations. I know that some people care about balanced equations, so I am going to try to balance my equations for people that do care. But like I said, for this course, the numbers are not as important as the concepts, okay? So remember, we are going to make 2-acetyl-CoA. And how do we make acetyl-CoA? We need coenzyme A. So coenzyme A, we represent as CoAsh. And I'm going to put little lines in there to remind me that those are bonds and that those are actual atoms. Okay, so coenzyme A is going to go in. We're going to make acetyl-CoA. We are going to, I guess this would be 2 if we want to keep everything balanced. And we're also going to emit 2 carbon dioxide. So I don't need my red pen because we did not make any ATP, but we did make 2 NADH. So how do we make NADH? We need NAD+. So there we go. And we're going to make 2 NADH. And I guess the last thing I need for this is a label. So this is pyruvate oxidation. Okay, so usually I tell people, take a piece of paper, turn it sideways, but you know, whatever works for you, because I do need a little bit of room on the right of that to talk about the citric acid cycle, and I'll add that in after we go through those lecture notes. Okay, so let's go back to our PowerPoint, and we'll talk about the citric acid cycle. So there it is. You can see we are emitting some more carbon dioxide. We're making some more NADH. We're also making an FADH2, and we're also making an ATP. And all those things can be multiplied by 2, by the way. So let's take a look at this here in a bit of detail. Guess that's the same picture. Apologize for that. Okay, so here's the details of the citric acid cycle. Another 10 reactions. The good part is we have this little thing here. right? So that citrate, if I were to put the hydrogens on, it would be called citric acid. So that's where the name citric acid cycle comes from, because we're going to regenerate that. And I know you're thinking, okay, big deal, citric acid. Well, if you like sour candy, that's the taste in sour candy, the citric acid. So it's a good thing. I like sour candy. Sour things are fun. So you can see the very first thing is the coenzyme A is just going to get recycled. So there it goes. Boom. We have our oxaloacetate here, and it's got 1, 2, 3, 4 carbons, and the 2 carbons from the acetyl-CoA join it, and we make a 6-carbon compound, which is citrate. And we make isocitrate, but who cares? Just some water going on there. Not really that important. What is important now is we're going to oxidize this thing until it is dead, like literally vaporized to carbon dioxide. So we're making some NADH here. So here we go. We're making another one down over here, getting rid of some carbon as carbon dioxide, of course. Another coenzyme A is getting used in there, which is not so important for this class. We have another electron carrier here, FADH2. I guess my writings just got zapped on there, but I'm going to summarize that up, like I said, on the whiteboard. And notice down at the bottom, we're making 1 ATP. So the truth is we're not actually making ATP. The truth is we're making GTP. So I know this is, confusing enough, but why would we make GTP instead of ATP? Well, if you think about it, ATP, A is the A from the genetic code. We actually have 4, right? We have A, we can make ATP, we could make GTP, we could make CTP, we could make UTP. ATP is just kind of, usually used, but sometimes GTP is used. And actually GTP can be used to make ATP, and it's equivalent energetically. So sometimes you'll see in the textbooks, they talk about an ATP being made. Sometimes they're more accurate. They're saying it's a GTP. This one, they're kind of doing a hybrid approach to saying that GTP can get converted to ATP right away. And I don't know why that enzyme is using GTP and GDP and not ATP. I have no idea. But sometimes nature does interesting and weird and wild things. Okay, so we do make 1 more NADH, and that happens way up the top. And the last step is the oxaloacetate is regenerated, and it's ready to make another round of citric acid. Okay, so end products, right? This is the kind of the synopsis of the whole thing. So let's add all those details to our whiteboard. Okay, so I'll share that with you. Here's the whiteboard. So glycolysis is just gonna, I don't think, or maybe I can shrink it a little bit more. I might have just enough room to keep that all on there. So this is still all in the mitochondrial matrix. And so here goes. I'm going to draw it like this, and then I'm going to draw a circle. So there's my citric acid cycle right there. So citric acid cycle. Okay. So I drew my arrow like that for a reason, because remember, we are recycling our coenzyme A. And if you want to keep things balanced, well, remember that there's 2 of them because we have 2 acetyl-CoA. So first thing, remember what's going on with all that carbon. We are basically releasing it as carbon dioxide. So how many carbon dioxide? Well, we have 4 carbon molecules left, so 4 carbon dioxides. Number 2 is the ATP. So we are making 1 ATP per round of the cycle, but we multiply that by 2 because, of course, we had 2 pyruvate originally. So remember that takes 2 ADP and 2 inorganic phosphate. So let's do that here and make 2 ATP. And then the last thing is we have those electron carriers, right? And so I have just enough room to squeeze some of them in. I'm going to make 6 NADHs. So there's 6 NAD plus and 6 NADHs. And then over here, I'm going to put my FADHs. And these things are not necessarily happening in this order, but like I said, this is more of a summary, right? So we have 2 FADs. Remember, that's an electron carrier. It's going to pick up some electrons and some hydrogens and become FADH2. Okay, so sorry if I just I didn't mean to go so close, but I'll just draw a line there to show you that they're separated. Okay, so I'll just give people another 10 or 15 seconds here to take a look at that. At this stage, if you take a look at this, we've got, let me find another color. Maybe I've got a highlighter here I can try. Right? We've got, it's not very bright, is it? Well, we've got 2 ATP here. I'll just color that in nice and thick. And we have 2 more ATP here. Okay, so that's not too bad. 4 ATP, you know, per glucose molecule. That's something. We can certainly do a lot better than this, though. And that is where these electron carriers are going to come in. So we've got some FAD over here. We've got some NADH over here. Got some more NADH over here. And all of these are going to be used by the electron transport chain. And we are going to make a whole load of ATP with them. And we're probably going to talk about that next day because I can see we're running out of time. So a couple more things about this, and then I'll finish up for today. No point in getting to the electron transport chain with a couple minutes left. So this is just the summary showing what we were just talking about. And this is kind of another summary slide of what I just mentioned, right? We have glycolysis. Glycolysis, we've made 2 ATP and 2 NADH. Pyruvate oxidation, we've made 2 NADH. Citric acid cycle, we've made 2 more ATP, a bunch more NADH, and some FADHs. And kind of like what I just said, not much ATP yet, right? But we are going to get there. And that's where oxidative phosphorylation comes in. So you can see we've got these ATPs here that have been made by substrate level phosphorylation. And now we're going to talk about oxidative phosphorylation. So I think I have a summary slide of this, which I'm just going to go through super quickly, and then we're going to review it in a lot of detail here next week. So take a look at this, right? Oxidative, that refers to the electron transport chain where oxygen is going to be used. And we have phosphorylation. That means we're going to add phosphates to ADP to make ATP. This is called chemiosmosis. Okay? Just take a very quick look at this whole thing. We've got these electron carriers being consumed. We've got hydrogens being put across a membrane. We're making an electrochemical gradient. These hydrogens eventually they go back into the mitochondria, and when they go back in, that energy is used to make ATP. That's all I'm going to say about it today. There's a lot here to explain, and it's just not worth it to go into any more detail than that for today. So Friday afternoon, I know I'm super generous. I'm giving you 2 extra minutes on Friday afternoon to enjoy your time. So hopefully you make the best of it. I will see everybody next week. Take care and have a good one.